All right, who's got some questions this morning? Sure. I don't think I put anything up for this week. We'll say we're done. See how simple that question. Well, one other thing I want to talk about with the prokaryotic gene expression, another sort of opposite event, like with the lac operon, you have what we would call a catabolic operon. So it's there to cause the catabolism of a food, a sugar. So catabolism is breaking down material. So the lac operon encodes enzymes that break down lactose, right? In the instance of those types of operons, you don't need those proteins unless that substance is around. If there's no lactose, then don't waste any energy making those proteins because it does take energy to do that. So you want them off unless that substance is there and you actually need it. If you have something else that you're already eating, glucose, yeah, you don't need to switch over yet. So that's where we see the regulation in the lac operon. Being a catabolic operon, it's inducible. For things like the trip operon, we're looking at building materials or synthesizing materials. So this would be an anabolism or anabolic side of things. So the cells also have the ability to synthesize amino acids and nucleic acids and all those things that they need, just like the yeast would be able to do. So the tryptophan operon is one of those that synthesizes the amino acid tryptophan. And it's a paradigm because there are many other operons that are anabolic that function in a very similar way. So with the trip operon, it actually has 5 different genes in it that are involved in the process, the steps, the biochemical steps for trip synthesis. It has a promoter and it has an operator, and there's a repressor that will bind to that operator sequence. So that's the trip repressor. And it's encoded by a gene, they call it TRPR for repressor. It's not located right next to the operon like the LAC-I gene was, which that's okay. It doesn't really matter where that gene is, but it's in the genome. So it makes a repressor protein that can bind at this operator. There's also a sequence here that is sometimes called the leader sequence, or your book calls it TRPL, that's fine. It is a short open reading frame near the beginning of the RNA, which is, it can be translated, but it can only be translated if there's plenty of tryptophan tRNA, okay? If you're running a little bit low on tryptophan tRNA, then it's going to take a little bit, it's going to stall here. And if it stalls there, that's it helps the RNA be made. It's called attenuation. That process, actually, is that going to pop up? That process is called attenuation. And we're not going to dive into attenuation because it's something that's pretty unique to prokaryotic organisms. Whereas these other concepts are used in prokaryotes, but they apply in eukaryotic organisms as well. So, really, is this thing locked up? Oh, yeah, attenuation. I don't know what it was doing. So, in the situation where you have this trip repressor, the protein itself, by itself, is not actually able to bind to DNA very well, if at all. And it needs the presence of tryptophan to actually stimulate that. So in the absence of tryptophan, the operon should be transcribed, right? If you don't have any tryptophan, dude, you better make some tryptophan. So in the absence of tryptophan, the repressor is non-functional. Okay, it's inactive. So at low tryptophan levels, now this doesn't mean you've completely run out of tryptophan. It just means you're running low on tryptophan, right? You wouldn't want to totally run out, right? You want to get gas when the little yellow light comes on, right? So when tryptophan levels get low, the tryptophan repressor is going to be just the protein. If tryptophan levels are more elevated, then you're going to get tryptophan interacting with the repressor, the little dots or tryptophan. And I don't actually know. I believe it takes 2 molecules of tryptophan, not 3. And the repressor functions as a dimer, not a tetramer. But anyway, it helps it form the dimer. And then that can bind to the operator. So this makes sense. If there's plenty of tryptophan hanging out and you don't need to synthesize your own tryptophan, then this would keep the operon turned off. So you're not wasting any energy making something you don't need. Okay, so at high tryptophan levels, the tryptophan interacts with the repressor. So we would call tryptophan in this instance a co-repressor. It's a small molecule that makes the repressor actually function as the repressor. So it's called a co-repressor. Okay, we talked about these different classes of molecules in one of our earlier lectures on this. So you have these two situations. Now, a lot of times they draw they want to simplify the diagram. So the promoter, remember, is the -10 and the -35, those two sequences that RNA polymerase binds to. And in this case, the operator actually is overlapping with those sequences. I have these out of order, but this is what it looks like. So, oh, come on. So here's the -10, and here's the -35. And this -10 is actually a pretty good one. And this -35 is actually a perfect match to the consensus. So the TRIP promoter is considered to be a fairly strong promoter. RNA polymerase does bind well, and it doesn't need any assistance for it to bind well, like the lac operon did. So all you need to do is just prevent it from binding, and that's where the TRIP repressor can bind. So there are 3 sequences that the TRIP repressor binds over. It reads all 3 of those and binds there, and that is right smack dab in the middle of where the RNA polymerase needs to bind. So it literally does just get in the way. The 2 proteins are not able to bind simultaneously, okay? So if the TRIP repressor is activated with the tryptophan, because there's tryptophan in the environment, it's going to be bound here, and RNA polymerase can't bind. So no expression, the gene is off. And if you're running low on tryptophan, then the repressor, the tryptophan that would be on the repressor is getting used in the cell to make proteins. Well, then the repressor is empty and it will not be able to bind to the DNA. And now that frees up your binding sites. So RNA polymerase has full access, and it will transcribe the operon, which encodes a bunch of enzymes that go right into making tryptophan. Well, what happens as soon as they crank out a bunch of tryptophan? Now the repressor can bind again. So it's a self-limiting thing. You're only going to make what you need for as long as you need it. So the TRIP operon is not the only operon that's regulated in this way. We find that all of the operons that encode things like vitamins, several vitamins, and all the amino acids that bacteria can make actually all function in the same way. They use a co-repressor, Okay, and the substance that the operon makes is the co-repressor, or in some cases an intermediate along the way. In this case, it's actual tryptophan, but in some cases, it's one of the biochemical intermediates along the way. So the other way that the tryptophan operon can be regulated is by that process called attenuation. And so I just only want to mention what it does. What happens if you're at not high, not an abundant amount of tryptophan, and you're not fully, completely running too low, but you have some medium amount? Well, you might want to make tryptophan enzyme every now and then, but you don't necessarily want to make a bunch of them. So attenuation kind of takes care of that middle zone, that middle area. So if we think about in the case of an amino acid in particular, think about where it can be in the cell, right? So you can have, we'll call it tryptophan F for free. It's just all by itself. It's just there in the cytoplasm, okay? You can have tryptophan that's part of a protein, right? It's already been connected in with other amino acids and it's in a protein chain. Where is it before it gets hooked into the protein, but when it isn't free? How does it get in the protein, right? Who delivers it to the protein, to the synthesis of the protein? Who or what? Think about translation. What part of translation machinery brings the amino acid to the ribosome? Again, something you can look up. But I really am worried that you don't know that right off the top of your head. Earth Life 1, UB 1, high school biology, watching the Discovery Channel. About 57 jillion times in here last week and the week before. What entity brings the amino acid to the ribosome? Huh? No. Yeah, got a 30% chance. Not gamble with them. Not the mRNA. Who? T. Yeah, the tRNA. You know, T for transfer. Right? So you have free tryptophan, and it can get connected to a tRNA that carries tryptophan, which could then take it so that it can get incorporated into a protein. So if you've got plenty of this, right? If you have a high concentration, you're going to have some free. If you're at a low concentration, right, you're somewhere over here. If you're at a medium concentration, you might not have very much of this, but you still have this, so you can still make proteins. And so here you want to start doing something about it. You want to start thinking about how much do I really have? Can I still make a protein? So what attenuation measures is essentially attenuation can sense the amount of transfer RNA that has tryptophan attached to it. Because attenuation happens where that little TRPL gene, it's just about 10 or 15 amino acids, but it tries to get translated and it has 2 tryptophan codons in it. And if it gets translated slowly, it means that you need some tryptophan. And so the rest of the gene gets transcribed. If it gets translated quickly, you don't need so much tryptophan, okay? So attenuation works by slowing translation, and if you slow translation, then you go on to actually transcribe the operon. Okay, that's really all you need to know. If attenuation happens, you get to transcribe the operon, or if it's slow. If translation is fast, then you stop or attenuate. Attenuate means to make less effective. So this, if the translation is really fast, that means you have plenty of this. Okay, if the translation is slow, it means you don't have very much of this, and you should probably make more. Okay, so slow attenuation is a way to either slow down translation or allow translation to happen at a regular speed. I can say it's fast. I can just say this is regular speed. Okay, this is like every other human. This is your grandmother. So this is testing out essentially how much tRNA tryptophan is hanging out. Okay, so attenuation is something that you find that is also used for all of the amino acids. Okay, all of the amino acids, because only amino acids are working in translation. You don't translate vitamins, and you don't, that, right? Those are synthesized. So they don't work with attenuation, but they do work with the repressor. And for tryptophan, it gets to work both ways. Okay, so really high tryptophan levels cause repression. If it's really high, you have repression. Okay, if it's medium level, you have some repression, but you also can have attenuation. And if it's a low level, you have no repression, and you have no attenuation. And so you get the proteins made. Okay, you have transcription. Now, attenuation can only work in bacteria because you need to have translation happening at the same time as transcription. And that doesn't happen in any other organisms because transcription happens in the nucleus and translation takes place in the cytoplasm in every other organism. Okay, so this can only happen in prokaryotes because there is no nuclear membrane, right? And so as soon as the RNA is made, you can try to translate it. So as soon as the RNA starts getting made here, it tries to get translated, it tries to start translating that leader. And if it can't translate the leader, it's all over, okay? Now your textbook goes into the mechanics of that attenuation. I don't really want to dive in there. It's kind of tricky and it's not really pertinent that much. I want you to know what attenuation does. Attenuation is a way of sensing the sort of medium level of material that might be attached to tRNAs, and it's functioning for operons like the trip operon, and it functions for the histidine operon and the alanine operon and the serine operon, all those operons, okay? They all work by attenuation. Okay, so we have our two paradigms, the lac operon, which is a paradigm for catabolic operons, and the trip operon, which is a paradigm for anabolic operons. So these are almost always going to be inducible and require an inducer to turn them on, and these are going to be repressible and require a co-repressor. Obviously, if you have a co-repressor, you have to have a repressor too, something to co with, okay? So that's kind of the general rule. So how are things different when we talk about regulating gene expression in cells that have nuclei, like us, eukaryotic cells, so yeast, the fungi, the protists, animals, and plants. So gene regulation in eukaryotes is quite a bit different because A, you've got a nucleus, you've separated the processes of transcription and translation in both space and time. You have RNA processing to think about. You have differences in the way the DNA is packaged in the nuclei that you have to think about. And you also have genes that need to be turned on only at certain times, which we saw with the lac operon. You only want it on when the lac is there. Trip operon, you only want it on when you need to make it. But with eukaryotics, multicellular organisms, you also have the consideration of different cell types. So cells become specialized to just do one thing. And you see that during the embryonic developmental period, you see different cell types arising and becoming specialized. So you have temporal and spatial expression regulation because you're going to have genes in different places at different times. Okay? Developmentally, you have expression of genes during the correct stages of the life cycle, during embryogenesis. Some are only during embryogenesis, others are expressed only in adults, some are expressed in both, but they're in different places when they're in one embryo versus the adult, things like that. And you have, of course, with the development of different tissues, tissue specificity, okay? Tissue specificity. So a gene that's expressed in only one tissue, we would call it a tissue-specific gene, okay? You have, like, neurotransmitters are made in the nervous system, but not other cells, right? Muscle cells make myosin and actin that are specific, and tropomyosin and things like that. So all of these different cell types are making a whole battery of proteins that only they make, and that's why they're that cell type, right? So how do those genes get turned on, and how do those genes then direct the development of that particular tissue type? So we had already talked about the basics of transcription. You need what we call general, or also sometimes called basal, either term, these general transcription factors, right? You have the need of them to get RNA polymerase to be recruited and brought to the DNA. So in bacteria, what did RNA polymerase do? It binds right onto the DNA. The sigma protein, the sigma subunit, recognizes two 6-base pair sequences, and it latches on. If it latches on really good, great. If it doesn't latch on so good, you might have to help it out. But in eukaryotes, the RNA polymerases don't recognize DNA at all. They don't recognize any specific sequence of DNA. Once they're brought to the DNA and they bind with the DNA, it's just, okay, it's a DNA. They don't care what the sequence of the DNA is. So the only thing that gives them the specific instructions to start at a certain place are the general transcription factors. Now, the general transcription factors may not be able to get access to the DNA because the DNA is packaged in nucleosomes, or they may need other assistance in recognizing the DNA and recruiting the RNA polymerase to that region. And so that's where you have additional transcription factors. We call them regulatory transcription factors. We also use the term gene-specific transcription factors. So these transcription factors would regulate the transcription of a gene just in the muscle, or just in the liver, or just in the neural cell, right? They are going to cause a gene to be tissue specific, right? So, and they may only work on one gene. So we call them gene specific transcription factors. They're regulating the process to give us those terms we just talked about before, spatial and temporal expression. Now, lots of genes are going to be expressed constitutively all the time, right? And they're going to be expressed ubiquitously. which means everywhere. Okay, why don't we just say all the time and everywhere? Because that's not cool. You want to be able to sound cool, right? You want to say constitutive and ubiquitous, and people are like, they're scientists, they're so cool, right? Well, that's what those mean. And we sometimes think, well, what's that? Well, every cell has to do metabolism, right? Every cell has to be able to repair its DNA. Every cell has to do transcription. Every cell has to do translation. So a lot of those genes that are making the ribosomes and the tRNAs and you know, the RNA polymerases and the sodium potassium pumps, all the machinery for just carrying out, you know, glycolysis and Krebs cycle and oxidative phosphorylation and all those things. Those are just ongoing things. Maintenance of this, you know, making plasma membrane, all that stuff. It just has to happen or you won't have a cell, right? And so we call them sometimes housekeeping genes because they're just running routinely to keep everything operational. Those are not going to be expressed very differently between muscle and liver or skin or whatever. All those cells all need to be alive and they all need to be doing those things. So those genes are not necessarily tissue specific, okay? Or differentially regulated in any way, shape, or form. They will be ubiquitously expressed constitutively, right, for the cells to be alive. So we're not going to really pay much attention to those genes in this category because they're not really under the same sort of control. They can operate just off the basal transcription factors. All they need is them, RNA polymerase, off they go, right? But these other genes, these tissue-specific genes, they need specific transcription factors. Okay, and so that's what a lot of study has been directed at, because it's when those genes go haywire a lot of times that we have pathologies. We have disease, non-functioning tissues or cancers, and things like that. So they've received the lion's share of the study. Okay. So in considering that sort of transcription, we've come up and realized that genes have different sorts of control elements. They have promoters and enhancers, right? So we've talked about enhancers and promoters before. A promoter is a sequence, any sequence of the DNA that regulates transcription. And when that sequence is moved or inverted, it no longer functions properly. Okay, so a promoter is orientation and position dependent. Promoter is dependent on its position and its sequence's orientation. When we say orientation, we're talking about if you read the sequence 5' to 3', the letters of the sequence. Okay, an enhancer is just the opposite. It's independent of those things. It could be moved. It sometimes can be moved a lot. Still works. It can be flipped around. Still works. Okay. Now, sometimes it might be able to be moved, but not flipped. Sometimes it might be able to be flipped, but not moved. That's okay. We'll still say that's an enhancer. All right. But a promoter, you just can't jack with it at all. It won't work. Okay. So things that are near to the start site are proximal. Things that are far away are distal, right? And enhancers tend to be more distal and promoters tend to be more proximal. But it's not an entirely cut and dried situation. There are exceptions to all these rules. So that just sort of lets us talk about where. Proximal, distal, okay? Means the same thing it does in any other situation, okay? So the sequences that are part of these, that the transcription factors recognize are going to be called elements. We've used that term already, but we throw some words in front of them. We might call them response elements or control elements or regulatory elements, or a lot of times you're going to have the word box added to it, like cat box or CE box or E box. Why box? What's that all about? Well, it's because, you know, when they were analyzed, people were drawing a box around it, and that just sort of caught on, just a joke, kind of, not really a joke, just a jargonal term. And as some things do, it just becomes the term, okay? So you'll see that, like Tata box, right? Tata box is a proximal promoter element. And then, as we saw with prokaryotes, that paradigm, you have repressors. So you have things that'll bind and prevent transcription, and you have activators, things that'll bind and stimulate transcription. And I would say that initially in the study of these things, activators were kind of the rule of the day. All the proteins that people were discovering that were functioning, all were turning genes on. Very, very rarely, in fact, I don't really know of any case that a differentially expressed gene is constitutively on. They're all constitutively off unless you need it or want it, and then they're turned on. So activation is more the rule than is repression, although we'll see some very notable exceptions to that, okay? So you have transcription factors that act as activators or repressors. We a lot of times don't distinguish between that. We just call them a transcription factor, okay? And you just kind of have to remember, you know, it's dark side, light side. Is it, you know, good Jedi, Sith, whatever. Is it an activator or a repressor? And then, of course, whether something activates or represses can also be how it's interacting with other transcription factors. So if a gene would normally be turned on by transcription factor A, but transcription factor B is also present, transcription factor B might just inhibit transcription factor A from getting to its position, then transcription factor B isn't actively repressing, it's just actively getting in the way and keeping A from working. And you're like, well, how is that not repression? Well, it is, but it's not because it's physically doing something to the DNA or keeping the RNA polymerase away. It's just not helping the RNA polymerase get there, right? It's passive aggressive inhibition, you might call it, right? It's not really keeping the RNA polymerase away. It's just not helping it get there. All right, so as we mentioned with the transcription factors like the LAC repressor and the TRP repressor is the same way, these proteins are going to have multiple functional domains that work in specific functions like binding to DNA, okay? So they are going to have, almost all of them are going to have a DNA binding domain that's going to recognize a specific sequence, right? That's an important thing. This DNA binding domain doesn't just let them bind to DNA, it lets them bind to DNA at a particular sequence of bases that they recognize. And that sequence of bases is going to be called their site, S-I-T-E, site. Binding site or box, right? If it's like a T box, okay, that's where that thing binds. It's an E box, that's where that thing binds, okay? They're also almost uniformly going to interact with other proteins. Most things bind as dimers at a minimum. Okay, some bind as monomers, and we'll talk about how they can do that, how they can get away with not having to have a partner. But most things bind as dimers. Sometimes two of the exact same protein, that's a homodimer, or perhaps two different proteins, which would be a heterodimer, okay? So this is very typical. They also are going to interact with other transcription factors and other proteins. So the dimer is physically binding to the DNA, but maybe that protein that's bound here binds to a protein which doesn't bind to DNA. So that protein would be interacting through some other domain. And so altogether, we call this a PID, or protein interaction domain. But don't be confused if for a particular protein, they say, well, this is the dimerization domain. Okay, if it's a dimerization domain, it's a PID, all right? Because they're just going to go ahead and be more specific about it being a dimer, okay? And then, unlike prokaryotes, eukaryotic transcription factors very frequently have a specific region of the protein that interacts with the RNA polymerase. And this would be known as its transcriptional activation domain, okay? TAD, or if it happens to be one that actively represses transcription, we might be able to identify a specific amino acid sequence that does that. And then that would be called a repressor domain. There's a very notable example in a protein called engrailed. Engrailed. First discovered in fruit flies, it's expressed in rings, which is what engrailed means, in rings. And it's, of course, every organism that has it that we've looked at, every animal organism has it, I know, maybe plants do too. But it specifically functions as a repressor, and there's a particular amino acid part of it that does that. And you can take that part off and you can put it on to another transcription factor that would normally activate, and it'll turn that one into a repressor. So it's actually a repressor domain. So that's an example of that. But those two domains would be, you'd probably have one or the other. You're not going to have both. And the vast majority have activation domains. Okay. So sometimes we just call it AD for activation domain. Sometimes transcriptional activation domain to be more in keeping with the three-letter abbreviation. But either way is fine. So what part do all of them have? They all have a DNA binding domain. And by an analysis of the mechanism or the conformation that the protein uses in that domain, we've used that to characterize them into families, okay? So there are many families, and within those families, there are even subfamilies. And so they're based in these families by the type of domains that they use to interact with DNA. And then a subfamily might be based on that DNA binding domain plus a second domain that they use to interact with something else, maybe a dimerization domain or something like that. So in this instance, one of the very most prominent groups, families, that has a very large number of transcription factors in it is known as the zinc finger family. Okay, so this is in parentheses is the approximate number. They have two subfamilies. They have ones that interact as a monomer and ones that interact as dimers. And the ones that interact as dimers are all steroid receptors. So these are things like the estrogen receptor and testosterone receptor and glucocorticoid receptor, right? When you have COVID, you're supposed to get some steroids. You're getting hydroxycortisone or corticosteroid analogs like dexamethasone or prednisolone or something like that. They're interacting with those steroid hormone receptors, okay? The glucocorticoid receptor, GCRs. I didn't list over here, but things like growth hormone receptor, estrogen receptor, retinoic acid receptor, the gene in Drosophila called CNRPs. These are all members of this subfamily. And then the monomeric ones, here's some members. The Wilms tumor 1, a gene, we mentioned that Wilms tumor, talking about mapping genes a long time ago. Well, it's a transcription factor and it happens to be a zinc finger. Now, why are we calling them zinc fingers? Well, we'll get into that. So don't worry about why we're calling them these things just right now, okay? Zinc's going to be involved. There is no finger, but they call it a zinc finger, okay? Another very extensive family are the homeodomain containing family, also sometimes called Hox genes. Just in general, they also are known as the Hox genes. But in reality, the Hox genes are one specific subfamily. There are 11. I only listed 4 of them here. But the Hox genes are involved in a lot of more large-scale anatomical events like, hey, that's the head end, and that's the tail end, and here's where a leg's going to be, and here's where a hand is going to be, and here's where a knee will be. They're doing these large scale things, like the Hox genes we talked about in the gene families we had. You saw how they were expressed from head to tail in these different regions. This is Drosophila gene, Drosophila gene, Drosophila gene. This is the engrailed gene that we just mentioned. And it has a Hox DNA binding domain, and then it has a repressor domain that goes with it. And then there's some other subfamilies. POW, that's pronounced POW, and it's a lot of times these acronyms are just the initials of some of the proteins that were discovered that all shared that domain. So the POW domain, PIT, OCT, and UNC. This is a human gene, a human and mouse. This is a human and mouse. This is a nematode worm gene. Mutations make them uncoordinated. They can't crawl straight, right? So the name of the gene is uncoordinated, or UNC for short. This one is expressed in the brain, so brain. This one is expressed in the development of the pituitary gland, so PIT. This one is expressed, this one is named because the sequence of DNA it binds to has 8 bases. It's called octamer binding, 8, right? But it's expressed in the immune system and a lot of other places, but the immune system is where it was discovered. The limb for these 3 proteins, limb 1, islet of Langerhans, and MEK, which is another nematode gene, mesoderm gene. And then there's the paired box, which is named from the fruit fly gene called paired. And then there's all these vertebrate homologs called PAX. So paired box in a vertebrate, and a lot of times you get just X added to the end of it. So PAX, we'll see that tradition comes in some other families as well. PAX makes your eyeballs. Without PAX 6, you don't have eyes. There is a family known as the basic. Okay, now basic doesn't mean like, oh, it's just your run-of-the-mill basic, t-shirt kind of thing. It has basic amino acids. It has positively charged amino acids in its DNA binding domain, and that's what it uses to bind to the DNA. And there are 2 subfamilies based on how they form a dimer. So these all work as dimers, and some of them form a dimer using leucine amino acids in their sequence, and they got the nickname leucine zippers. And then other ones have an amphipathic helix. That's a helix that is charged on one side and very nonpolar on the other side. So the 2 nonpolar sides want to go together. And it's actually 2 helices like this that are charged on the outsides and nonpolar on the insides. And you have 2 other ones just like it, right? So how do they get together? They actually kind of like dovetail together. So all of the hydrophilic parts are on the outside and the hydrophobic parts are on the inside. And so that's called a helix. That's a helix. Loop, helix. And then they come together. We'll look at the pictures of these. These are things that are found involved in, like this one is what makes muscle cells. You can take this protein, express it in a skin cell, precursor skin cell called a fibroblast, and it'll turn that cell into a muscle cell. That'll grow on a petri dish, and then it'll start contracting. So yeah, it's a muscle cell. And so that's called myodifferentiation, MyoD, muscle differentiation. And these are several Drosophila genes that work in the same sorts of things for the nervous system. Here, these are all for the nervous system. These are transcription factors that are involved in a lot of really early tissue decisions to, go down a lineage, like to go down to be neural crest or to be mesoderm. Early in embryogenesis. The Fox family, okay. So there was this protein in fruit flies called fork head. And so all the members of this family have that same little domain. And after they started finding members, they said, it's a fork head box. Fox. That sounds a lot better than fork head box. So there's fork head. That's a fruit fly gene. That's a human liver gene, turns on genes in your liver, hepatocyte nuclear factor 3A, 3 alpha. It's not a huge family. As you can see, the numbers are dwindling down here. The SOX family, the original member of the SOX family, gentlemen, is what made us able to be gentlemen. It doesn't make you a gentleman, but it makes you male, and you may choose to be a gentleman. So the SRY gene, sex determining restricted to the Y. That's what that stands for. And then they had an SRY box and these other proteins, and they said, oh yeah, SOX, SRY box, SOX, right? And that's all because of this original homeo box, HOX. So SOX, PAX, FOX, all these things, okay? And then the last one, there's not a whole lot of them. It's MADS box. In fact, the acronym pretty much covers all of them. So there's a muscle regulatory gene called MEF1. This one is a yeast gene that's involved in switching between the two sexes, alpha and A cells. And this one is a plant, that one's plant, and that one is human again. And so that's what that stands for. It stands for MEF, MCM1, A-gammins, deficiens, SRF. And so they just threw all those letters together and it made a MADS, so they call it MADS. And there's a whole nother family of regulatory proteins called SMADs, okay? So don't confuse them. These are transcription factors and those are transcription factors, but they're not the same kind and they're not a family. All right, so how do these things work? Yeah. How do they work? Well, DNA binding domain, it's binding DNA. Well, how does it bind DNA? Well, it's going to form some sorts of hydrogen bonds with the bases of the DNA, or it's going to interact maybe ionically with the backbone of the DNA, the phosphate groups, which are charged. So zinc fingers were named zinc fingers because there's an atom of zinc that is important to chelate with the protein and hold a conformation. And that conformation consists of an alpha helix, and the alpha helix has the amino acids on it that like to interact with the bases. So this yellow alpha helix here can interact with the DNA bases that are right in this groove. And the zinc finger proteins that interact as monomers, remember there was a group that were monomers, they all have at a minimum 3 zinc finger helices. So in this example, you have a zinc finger, I don't even know which one, oh yeah, ZIF268, that's its name, zinc finger protein 268. They're like stars, there's so many of them, they just like give them a number. So it has 3, right? And each one of them kind of lays into the major groove of the DNA, right? This one over here has 4, lays into the major groove of the DNA. So it basically just spirals around the DNA, and that's how it latches on, okay? So to latch on as a single protein, it seems to be that you have to wrap all the way around the DNA, okay? And so that one can do that. Otherwise, you're going to need to wrap all the way around the DNA, but if you can't do it as one protein, you're going to have to interact with another protein, or basically kind of grabbing it from both sides that way. So to hold on to the DNA, it seems like you have to bind into major grooves on both sides of the DNA. And this is binding into the major groove in four places, here, 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 and here, okay? All along the axis of the DNA. So that's how a monomeric zinc finger would work. In this example, this monomeric zinc finger, you can see that they looked at, well, what bases are there in those positions where each finger is interacting? And they found that the sequence is G, CG, T, G, 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 C, G, right? This one interacts with 2 amino acids, in particular arginines, and this one interacts with a histidine and an arginine, and this one interacts with 2 arginines. This finger, this helix, and this helix, this represents the zinc this represents the helix, they have same amino acids, or are at least positively charged amino acids, right? Remember, histidine can be positively charged. So these amino acids are interacting, and it seems like they interact with the G's, which they could, because the G's have a hydrogen bond there that would interact with them. Okay, so here's another protein, but it's in the same family. It's called CROX20. It's a chicken gene, and it's important in making a part of the brain we would call our brain stem for the chicken. The chicken calls it its brain stem too. So it has 3 fingers, and those 3 fingers each bind with DNA sequence, and that sequence is shown here. The first finger binds to the sequence GCG, the next one has GGG, and the next one has GCG again. So one thing we kind of notice about zinc finger proteins is they tend to like to bind to G's, and GC-rich sequences. So here's the amino acid sequence of each of the fingers that interacts. So the first one has this sequence. The second finger, which is a little bit further down the protein, right, like here, has this sequence, and the third one has that sequence. Now you can see a lot of the same exact amino acid is present every time, right? But this one has GCG, GCG, and if you look, these two have the same amino acid here and here, and a different amino acid in the middle finger, which has a G there instead of a C there. So some smart postdoc or grad student or something said, I wonder if that's why it binds to a different sequence. And they said, oh, we could fix that. So they did essentially a compensatory mutagenesis. They said, dude, mutate that to a C and ask if it works, okay? So if you mutate this to a C, that will not work. I don't know where my arrow went, but it will not bind. And they said, okay, well, now change that H to an E. Make the protein that has a glutamic acid there instead of histidine there. Sure enough, when you make that change, this protein will bind to this altered sequence, but it will not bind to the original sequence. So they kind of said, okay, so that part of the protein is kind of important. And they changed the T as well. So those two amino acids are the 2 amino acids that are critical. This led into an entire industry, okay? People said, we can engineer a protein to bind pretty much any DNA sequence. So you give us a sequence, and we'll be able to maybe figure out a zinc finger that'll bind to that sequence. And these became known as zinc finger binding proteins, and a whole industry established on it. Because what you can do is you can have the DNA binding part, the zinc finger part, but you can attach that to anything you want. So you could attach it to a fluorescent protein and use it to just tag a piece of DNA. You could attach it to a nuclease and have it cut the DNA. And that's what they really started working with. Those are called zinc finger nucleases, ZFNs. And they started using those to try to target specific genes to try to do specific mutations in the genome and edit the genome. Like if you had a gene that was acting up, was being expressed, causing a cancer, maybe you could just target that gene and eliminate it. Cancer done, right? So the idea that a specific sequence would interact with a specific set of bases is pretty cool, and that you could actually manipulate it to make it interact with the base you want it to interact with. That's pretty cool, okay? The other zinc fingers are all found in members of the steroid hormone superfamily or subfamily, and they all bind as dimers, okay? So the zinc finger is really just a zinc atom that is causing the protein to form in a particular conformation. I guess I don't have it on here anymore, but there are 2 amino acids that can interact with zinc real easily. And if you look at these zinc fingers, they can be characterized based on I mean, you can tell it's going to be a zinc finger if you find these amino acids in these positions. So, so if you find a cysteine and a histidine about 4 amino acids apart, and then any distance, doesn't matter, and then you find another one, and then you find another one, right? You either find 2 or you find more than 2, or you can find ones that are just cysteine, okay? And I think they're also about 4 bases apart, 4 amino acids apart. So if you just search the amino acid sequence and you go, oh, boom, boom, cysteines, you're going to go, ah, that might be a zinc finger, right? So you can just build a computer algorithm that'll just say, look for that. And it'll go through and go, oh, found one, found another one, there's another one. You know, there's a whole genome sequence, look at all the proteins and find them. Okay, and so they've done that, and they estimate that there's a crap ton of them. Okay, now that doesn't mean that every time you find it, it really is a zinc finger. It fits the pattern. It doesn't mean it actually does. Okay, and the reason is because these can form chelation with zinc. Right, this thing would bend around, and those cysteines, which have the sulfur on them, right, can form a loop with the zinc. Right, and that can hold that protein into its particular conformation. And in this case, one of the zinc fingers creates a dimerization domain, and the other zinc fingers create DNA binding domains. So the zinc finger that's the DNA binding domain is how it got in the family. This zinc finger is actually its protein interaction domain. Okay. Now these are steroid hormone receptors, and they also then have to bind to a steroid hormone. Okay. So you recall that one of the domains we had in the lac repressor was the inducer binding domain, or we called it a ligand binding domain. These also have a ligand binding domain for the steroid. But the steroid hormone does not affect their ability to bind DNA. It almost always affects just their ability to get into the nucleus. Okay, so these proteins are usually found confined to the cytoplasm because they're going to be interacting with a chaperone, another protein that hangs on to them and keeps them from getting into the nucleus. But when the hormone enters the cell, right, and these are steroid hormones, so they just go through the membrane. When they enter the cell and they bind to the receptor, they change the shape of the receptor and make the chaperone turn loose. And when the chaperone is off, now these things can be transported into the nucleus where the dimer, which is already together, and it still has its hormone on it, but it can bind DNA. It's not technically the hormone that's binding the DNA. And we can prove that by just eliminating the chaperone protein, and these things all go to the nucleus and bind. They don't need the hormone. The hormone is just making the chaperone turn loose, okay? So most of them can form both homo and heterodimers, and when they form those different dimers, there's a huge family of proteins that they can choose from to bind with. So this shows some of the family members. So you have one called the RXR. It binds to a retinoid. It could bind to retinoic acid or retinal or retin-9-cis retinol and all these different retinoids. There's a whole bunch of them. You have the RXR and the retinoic acid receptor. Retinoic acid only binds retinoic acid. You can have it with the vitamin D receptor, which binds to vitamin D, which is steroid hormone. You can have it bind to the thyroid hormone, which is thyroid hormone receptor. I don't know what that is. And so you can have, and there's more than one RXR. That's why there's an X in the middle. There's more than one of them. So you can have all these different interactions. And in this case, they bind to two sequences that are spaced apart by one base. Here they're spaced apart by 3 bases. Here they're spaced apart by 2 bases. Here they're spaced apart by 4 bases. Here they're spaced apart by 5 bases. But it's exactly the same DNA sequence. So the arrow represents exactly the same DNA sequence. I don't know, it's not, I didn't put it on the slide. No, I didn't. But it's just spaced differently. So if you want to activate a gene with those sequences spaced 5 base pairs apart, you can only do that with this combination. This combination won't do it, okay? So it's kind of important. So they're looking for different length binding sites in order to activate genes. So this would activate genes in your thyroid and things that are activated by thyroid hormone. This would activate things that need vitamin D to be activated, whatever those are, right? So they are going to all interact with slightly different sequences. So this is just the retinoic acid family, right? All of these things are actually similar to retinoic acid. Glucocorticoid receptor works the same way, except its hormone is one of the glucocorticoids. It could be cortisone, hydroxycortisone, prednisone, dexamethasone. All of those chemicals interact the same way. Okay, our other family, the homeodomain, another really huge family of proteins, and they tend to interact as individual proteins to bind DNA, and then they interact with a totally different protein to coordinate that binding. And we're not really sure if it's that protein helping the homeodomain protein or the homeodomain helping that protein, but they tend to be not really a dimer, but one interacts and then the other one interacts. So it always works as kind of in a partnership, but it's not really forming a dimer. These things can bind to DNA all by themselves because they have really 3 DNA binding domains in one. They have a helix that interacts with the major groove of DNA. They have another helix, which tends to interact with the backbone of the DNA. They have a third helix, which again interacts down in the major groove of the DNA. So this was known as a helix turn helix when they thought there were just 2 helices doing this, and then they realized, oh yeah, this other one helps. So now it ought to be helix turn helix turn helix, or helix turn 3 or something like that. I mean, they ought to come up with something more clever. This is the DNA binding domain that the LAC repressor uses. This is the DNA binding domain that the TRP repressor uses. This is the DNA binding domain that over half of prokaryotic DNA binding proteins use. So it's considered to be perhaps the most ancient DNA binding motif, since it's in prokaryotes and it's still in us, and it's in plants, it's in everything. So the homeodomain is 90, these 3 helices constitute 90 amino acids. So the homeodomain is a pretty concise chunk of the protein, and they'll be contiguous 90 amino acids. They won't be separated up. Okay, zinc fingers can be separated apart by quite a bit. So here are some examples. So the LAC repressor here forms a tetramer, right? So it forms a dimer. The dimerization domain is in this kind of the big chunk of the protein in the middle, and the tetramerization domain are through these amino terminal helices down here. And the DNA binding domain is through the carboxy terminal helices that are over here. You can see each protein has one, kind of back here, 2, 3 helices. Okay, and that's its DNA binding domain. And each DNA binding domain binds as a partnership to the entire operator. But if you look at the operators, they can be divided into what are called half sites. And if you read the half site here, 5' to 3', and you read the half site here, 5' to 3', they're almost identical. So we might call that an inverted repeat. So the half sites are imperfect inverted repeats. So that's how they bind. And this is a bacteriophage protein that does the same thing. It binds, here's helix 1, 2, 3, okay? Here's the engrailed protein. It has helix 1, 2, 3. So homeodomain proteins are fascinating proteins. They were first identified in mutants of fruit flies where parts of the body completely transformed into a different structure. Like in the case of the gene Antennapedia, or the mutant Antennapedia, the antenna developed as legs, not normal. And in the case of ultrabithorax, where the structure here, if you look closely, you can see this little nub of a thing right here I'm circling. Okay. For those of you pretending to watch at home, right here. That little thing is called a haltere. And in this case, you don't have a haltere, you have another wing. So it turns that segment, the third thoracic segment, into an entirely second segment, an additional second thoracic segment. So you have 2 thoracic segments, 4 wings then, okay? Like you would find in a dragonfly or butterfly. You basically turned a dipteran into an orthopteran. Well, that's called ultrabithorax because, hey, 2 complete thoraxes. This is Antennapedia, antenna leg, okay? These were called homeotic transformations. You've turned one structure, the antenna, into a completely different structure, or the haltere into a completely different structure. And so they started looking for the mutations, they mapped the gene, they finally cloned that sequence of DNA, and they discovered that these DNA sequences are all next to each other on one of the chromosomes, chromosome 3. And that gave us our Hox gene family, and we talked about that. So the homeodomain proteins all are going to have that same sequence, but the Hox genes in particular are the ones that do this cool kind of stuff. Okay. All right, the basic helix loop helix or basic zipper proteins. They have the same DNA binding motif, but they have 2 separate dimerization motifs, and they all work as dimers. So this shows the basic region is a simple alpha helix that has amino acids that are always either arginine or lysine that will interact with the major group. And so they've been kind of described as maybe like a clothespin clipping onto the DNA on both sides. And then sort of the spring part of the clothespin is this set of helices that are amphipathic. Amphipathic means you have 2 different charges at the same time. They have positive, or they have hydrophilic amino acids on one side of the helix and hydrophobic on the other. Amphipathic. Okay, like amphibian. And these are some different ones. And this is the basic sequence, the basic domain. You can see it has arginine, 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 bunch of arginines, sometimes histidines, sometimes lysines. K is lysine, okay? And then there's a helix 1, where some amino acids are polar and some amino acids are not. And if you just look at it, you're like going, how do I know? Well, you have to know your amino acids. But there's another way of looking at it in what's called a helical wheel. So if you realize this is forming an alpha helix, think about this as a spiral. You're going to start at the back of that spiral, and you're going to start with the first amino acid in that spiral and go here, okay? Then you come forward one amino acid. It's going to be over here. You go forward one more, it's going to be up at the top. Go forward one more, it's going to be back over here on this side. Go forward one more, it's going to be down on the bottom. So they every 1, 2, 3, 4, 5, 6, 7th amino acid will be or so will be on the same side again. So what they see, okay, look, polar, 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 polar charged, polar charged, polar, 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 not polar, not polar, not polar, polar charged, polar charged, polar charged, charge, 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 polar, charge, charge, charge. So this is hydrophilic on this side. This is all leucines or an isoleucine, all hydrophobic. Hydrophobic on that side. So this part wants to be next to something else that's hydrophobic, and this part says, yeah, whatever, water, you know, another protein that has charged residues, I don't really care, just as long as I don't have to be next to a hydrophobic one, okay? So that's called a helical wheel representation. So like these leucines are that leucine, then that one, then that one, then that one, then that one, then that one. Of course, that's not a leucine, but it works just like a leucine. So a helical wheel representation of a protein gives you more of an idea of where does the amino acid end up in the actual alpha helix, okay? So on these bases, right, you look and you're like, okay, every 7th one is the same type of amino acid. So you got an amphipathic helix. And then the leucines all being on the same side of the protein, somebody thought, that seems odd that it's leucine every time. It could be any there's a whole bunch of aliphatic amino acids that are hydrophobic, right? There's a whole bunch of them. So why is it always leucine? That's probably not a coincidence. There's probably a reason it's leucine. So they started mutating them and sure enough found out, yeah, if you don't have leucine there, it doesn't work so good. Or if you don't have something that's really similar to leucine there, it doesn't work very good. So you can't put tryptophan there or something like that. It doesn't work. So they came up with the idea that these leucines are actually interdigitating, perhaps, like the teeth of a zipper, okay? Whether they really do that or not is not really very clear, but clearly they do need to be leucine. So it caught on, and so they're called a leucine zipper. They have a basic domain here, and a leucine zipper in the other part of the helix, right? So here's one, a whole slide with a whole bunch of different ones. They're in this family. And you go leucine, 1, 2, 3, 4, 5, 6, 7. Leucine, 1, 2, 3, 4, 5, 6, 7, 7, 7. Every 7th amino acid is a leucine. Or something that's really similar to leucine, like valine or methionine, but most of the time it's mostly leucines, okay? This one's weird, okay? Boom. Probably not the leucine zipper. Probably the leucine zipper only has 4 teeth on it, on that one, right? So here's the idea of how they could interact. Leucine, 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 making some sort of hydrophobic interaction. Great. So these are called B-zips, and the other ones are called BHLHs. So B-zip, BHLH. The DNA binding domain is the B, and this part is the dimerization domain. So you're a B-zip or you're a BHLH. Yeah, there's a couple of proteins that are like, yeah, we want to, you know, play both sides of the game. We'll be BHLH zips. So they have a helix, loop helix, and then up here, they have a leucine zipper. They're like, it's all good. It's all good. And they all form heterodimers. Every protein that has this forms a heterodimer. And Mick and Max, you've met them. You can call them Mick and Max. We call them C1 and R. These are what the 2 proteins in corn, they're transcription factors, and that's why we talked about how they had to form a dimer. So like C1i has all of this stuff, but it's missing its basic domain. And so it dimerizes, but it cannot form a functional dimer. The MADS box binding proteins. They're in a world all on their own. They interact with DNA, and when they do so, they cause the DNA to be bent. They contort the structure of the DNA. They were originally called architectural transcription factors because they changed the architecture of the DNA chromatin structure. They would bend the DNA, they would disrupt the position of nucleosomes, and therefore allow the DNA to have access to other transcription factors. So that's kind of their role. And they really don't have a defined sort of structure that interacts with the DNA. They have a helix, and then the N-terminus of the protein has amino acids that kind of fit into the minor groove of the DNA. So this is the minor groove. Not many proteins bind in the minor groove. It's a smaller little slot to fit into. And most of them are over here in the major groove. Well, it has this little tail that fits in the minor groove. And that makes the minor groove need to open up a little bit. And if you do that, that causes the DNA to sort of twist a little bit, and that is what makes the whole thing curve. So they have a dimerization domain or a protein-protein interaction domain, which is a series of beta sheets. That's kind of weird. And then they have this DNA binding domain that doesn't really have a particular shape to it. It's more or less just a tail of the protein. It does have a similar sequence of amino acids, and that's why they're all in this family, this MADS family. SRF gives us a chance to talk about these acronyms. S is for serum, and RF, when you see RF, usually it's R is for response, and F always is for factor. So serum response factor. When you have cells in a petri dish that you take out of an organism, like if you took some skin cells from a tissue sample or something and you put them in a petri dish, they just sit there. They don't go through mitosis, okay? But if you give them certain factors, they will, okay? And one of the factors that they found was isolated from serum, right? They first just took serum, like when you donate plasma, that's serum. They just, put that on there on the cells, and they started dividing. So then they took the serum and they started isolating and separating all the proteins, and they found this one protein that just by itself caused them to divide. They called that serum factor, and then they found the DNA sequence that responded to that or was necessary for that. They called it serum response element, SRE, and then they found the protein that actually binds there, and they called that serum response factor, okay? So again, it's just throw something on a cell, see what happens, and they had mitosis as a consequence. And that's why almost all these things called growth factors are called growth factors, because they isolate them in a similar way. When they would put them onto cells in a petri dish, the cells would divide, grow, or stay alive. So you have nerve growth factor, epidermal growth factor, fibroblast growth factor, and then things like serum response factor and some of these other proteins. So these were proteins that were originally found because they cause DNA to replicate in the cells to go ahead and divide. And that's what they do. All right. Well, I guess that's kind of the end. So next week we'll talk about the activation domains, and they're fun as well. It's all about transcription factors. You guys ever see the movie Fletch? Chevy Chase, a long time ago. Definitely worth it.